Hello, my name is Michael Lawson. I'm head of product at Singular Genomics. I'm very excited to bring you this update on G4X. If you're new to G4X, I think you'll be very excited to see the quality of our data, the multiomic readout we have with H&E, protein, and transcript all from the same section, and the scale at which we're able to generate this data. If you're familiar with G4X, it's not your first time seeing one of our talks, I think you'll still be very excited to see our updated product offerings and our new data release. So no matter which side you fall on, I think this will be a very exciting talk for you. Now, if you're new to G4X, the main thing I hope you take away from this talk and this spatial summit more broadly is the massive throughput and cost savings that come with G4X and how they're paired with impressive performance and integrated multi-omic readout. And finally, I'll talk a little bit about a novel capability that's still in R&D phase. In addition to the unprecedented throughput, G4X also provides a high level of flexibility. For example, in the 4-lane format, you can run 4.5 by 4.5 millimeter sections. You can run 8 of these in one lane. You can run 32 of them in one flow cell. And because there's 4 flow cells per run, you can study 128 different samples in one run. In addition, each of these lanes are individually addressable, and I'll show you an example of what this can look like and the power it provides later in the talk. If you're interested in larger area per section or per sample, we have our 10 by 10 millimeter format. This is the 2-lane format. It allows for 10 sections per flow cell. So that's 10 centimeters squared per flow cell, or 40 centimeters squared, or 40 samples across an entire run. This presents an order of magnitude higher area than you get on any other platform in the field. In addition, while the G4X is a relatively new product, it is built on the G4, which is a mature instrument that has been in the field for a few years now. The high throughput is mediated both by this high density of tissue samples, which I'll explain how we manage that later, and our ultra fast imaging, which gets 2 billion pixels per second. We have developed curated disease-specific immuno-oncology panels for off-the-shelf use by our customers. Of course, we recognize each customer's application will be slightly different, and therefore customization is of paramount importance. We therefore constructed our panels in a modular way that enables customization at multiple levels. So I'm going to walk through how our curated panels are constructed, and then from there I'll talk about the different ways in which we can bring in customization. Each of these panels is built on a base of about 200 immune and stromal cell markers. On top of that base, we build in greater than 100 tissue or disease-specific markers, and in this case, it's for kidney cancer, or for lung cancer, colon cancer, or for breast cancer. Now, if you're interested in one of these 4 disease types, and you're happy with most of the markers we have, but there's a couple of genes of interest that we haven't captured specific to your application, you can very easily add on 50 user-selected or custom genes on top of one of these panels. On the other hand, if you're studying a tissue type that is not one of these 4 tissue types, and so you're not interested in any of these add-ons here, you can define your own 150 gene add-on to that 200 base immune and stromal base. Finally, if you're not interested in any of these markers, or you're working in an organism that isn't human, we support full customization of up to 500 genes. Now, which level of customization is right for you will depend on your application. And so we really recommend that you reach out to us and work with our team so we can get the best solution for your work. Finally, before I move on, I just want to give a little advertisement for something that will come later in the talk that will give a sneak peek of some new panels. One is human pancreas and the other is a mouse atlas panel. As I mentioned on the previous slide, in addition to our 4 curated disease-specific panels, we allow customization. This customization allows us to go beyond those 4 tissue types to a wider range of tissue types. And in fact, that's what you see here. Now, these samples get run through our Technology Access Services Lab, and you'll see that data in some of the other talks during this spatial summit. But what I really want to highlight here is that we've had great success across a range of human and mouse samples, and in fact, even with TMAs. And one of the reasons for the success is we have developed what we call tissue grit, which is a surface coating for our glass slides that allows us to get good adherence across all these tissue types, including difficult tissue types with lots of adipose tissue like breast. So you get this beautiful data. These are transcript maps paired with H&E maps. And again, we're even able to do this with TMAs. So if you want to take one of our highly plexed 32 sections on the slide and make each one of those TMAs, you can get to even higher plex in one run. As I mentioned, we have a multiomic assay. So in addition to those transcript panels, we also have protein panels. Our protein panel is constructed of 15 IO markers, including important markers such as CD3, CD4, CD8, and FOXP3, Ki67, PD1, PDL1. In addition to that, we have isotype controls, and we have 2 slots for custom targets. This allows you to build customization specific to your tissue, disease, or study as needed. Now, I'm going to spend more time digging into data once we get past this initial introduction to G4X, our panels, our workflow. But I just want to pause for a second on this image here from a human tonsil. I'll provide more zoom-ins and more data later, but already here you can see very nice specificity of CD20 in these germinal centers, just beautifully blue, with the occasional T cell inside, which is expected. And then outside, you have these T cells in green, red, and magenta, whether they're CD4 or CD8 positive T cells. So you see great specificity, but beyond that, you can already see, even at this level, and I'll provide further zoom-ins later, you can see beautiful morphologies, beautiful rings around the cell. And so in this multiomic assay, we're getting 17-plex protein along with 300 plus transcripts, along with H&E, all from each section at this high throughput. We're providing immunofluorescence quality protein data. And you'll see more of that as we go along. The G4X's unparalleled throughput is largely mediated by our novel tissue handling method and associated tooling suite, TissueStamp. I'll briefly walk through this workflow and some of the advantages. But if you missed Giannis's talk this morning, I highly recommend going back and watching the recording to see what this workflow looks like out in the wild. In addition, I highly recommend sticking around later today for our spatial histopathology session. The workflow begins with the standard section of a 5-micron FFP section and floating it on water. However, we catch a section on a stiff hydrogel pad rather than a glass slide. In standard histopathology workflows, and those for other spatial platforms, it is essential that you catch a section in just the right spot on the glass slide the first time, which requires a lot of experience and a lot of expertise. In our workflow, you can catch a section anywhere on the pad, and if for some reason there's a problem when you catch it, for example, a fold in the tissue or the tissue overhanging the side of the tray, you can easily refloat the section by submerging the tray back in the water and trying again. The next step of the workflow highlights why the exact location where you catch a section does not matter. The hydrogel pad is not the final surface that the tissue will adhere to, but rather an intermediary so it can punch out the region we care about. In addition to removing the need for precisely catching a section on a glass surface, the punching also removes the need to score your block before sectioning, which inevitably damages pressure tissue deeper in the block and often results in poor section quality. Finally, we place the cutters in this array, which when full is covered with a glass slide. The full setup is then flipped, placed on a heat source to complete the transfer. The array allows us to easily and precisely place our tissue at high density, maximizing our imaging area. Finally, samples can be stored and shipped on the hydrogel slab or on the glass slide, which removes the headache of getting permission to send full blocks between labs. You can instead easily section and pass those along to the core facility running the G4X. Taken together, the TissueStamp workflow lowers the barrier for entry to spatial multiomics on FFP blocks. The G4X is built on NGS chemistry, and so provides true in situ sequencing where we read out bases in space. We use this for both transcript counting and protein detection. For transcript counting, we bring in a Pavlov probe that hybridizes specifically to RNA in such a way that the two ends are in close proximity, forming a nick. That nick can be ligated and the rolling circle amplified by RCA and read out by sequencing. Now, our approach is barcode free. And so what we're reading out is the actual RNA sequence, which confers a number of advantages, some of which I'll highlight on the next slide. The small target area means we get efficient capture even in degraded samples, such as FFPE or bone marrow, where the decalcification process can greatly degrade the RNA integrity. In addition, if we have an RNA target that is either low abundance or extreme importance, we can titrate up the number of probes for that RNA target and increase the sensitivity. Finally, we get submicron localization, and by using multiple primers, we can detect RNA arbitrarily close together. Now, we have a future offering here that I'm not going to spend a lot of time on called DirectSeq. And what this allows you to do is read out a previously unknown sequence because we are, in fact, reading out bases. This is not possible by any method that has a hybridization readout. We're very excited about a number of applications here, from optical pooled screening, where we read out a CRISPR guide RNA, to reading out fields of single nucleotide variations, and of course, immune repertoire profiling. More to come in that in the future. Oligo-conjugated antibodies are used to detect our proteins of interest in our assay. A padlock probe is then brought in to hybridize specifically to that oligo. Once ligated, it provides a template for RCA. This amplification process greatly increases our signal above background, removing the need for any autofluorescence quenching. We then bring in a primer to a single base extension to identify 4 proteins, 1 per base, ATCG. Performing a single base extension and then switching primers for protein detection reduces the possibility of bleed-through between different antibody targets. Our native probe level data allows very efficient, very easy panel refinement. We use this approach to optimize our disease-specific curated panels I spoke about a few slides earlier, but it also allows us to very quickly, very efficiently optimize custom panels for your applications. So to give you a sense of what this looks like, we'll take the example of this gene C1s. In this case, we have 12 probes for C1s. And if we look at what these probes are doing in this tissue type, I don't need to tell you what tissue type this is. I don't need to tell you what gene C1s is. You don't need to know anything about either of those. But very quickly, you see that all these probes have a very distinct spatial pattern. There's a low level of expression across the tissue, but then high enrichment in whatever structures are towards the top left corner here and then across the top. See that with almost all of these probes, except 2. Those 2 probes have a low level of detection across the entire tissue that looks unspecific. Now we can identify this very easily and very quickly by eye looking at these images, but that's not going to scale to a panel level. So what we do is we calculate Ripley's K-statistic. And in short, that just allows us to do pairwise comparison across each of these probes for spatial correlation and see which probes are not behaving like the rest. So in this case, very quickly, the statistic tells us probe 5 and probe 9 are no good. Now this makes it very easy to optimize our panels, swap out probes for improved probe design, but it also allows us to not waste that initial run. Because we know which specific probes are misbehaving, we can throw that data out bioinformatically and recover the data from that run, as well as using that information for further optimization. And this is something that doesn't exist anywhere else in the field currently. So we're very excited about this in terms of its ability to support fast, efficient, high-quality refinement of custom panels for your application to get you the best data possible. Now that I've introduced you to G4X in our assay, I want to give you a sense of what this data looks like at scale before digging into specific examples. So in this case, we're looking at one 10 millimeter by 10 millimeter, so 1 centimeter squared, 5 micron thick FFPE section of kidney cancer. Now this sample is really beautiful because it's right at the transition between a healthy zone, where we see the expected structures, which we'll see when we zoom in and see subcellular localization, and a tumor region here indicated in green with a transition zone in between. So once we zoom in, we can see the expected structures in kidney. We see the glomerulus, we see these intermediate tubules, collecting tubules, distal tubules, all the structures you'd expect to see with subcellular localization. But now we can zoom out to the scale of that one 10 by 10 section. And as I mentioned earlier, we get 10 of those 10 by 10 sections, or 10 centimeters squared of tissue in one run. These may look similar, but in fact, they're serial sections, and they're here to highlight that throughput. Now, that is what you get from one flow cell. And in that one flow cell, we get not just transcript information, we also get protein, and we get H&E. So now for 10 centimeters squared, we have protein, transcript, and H&E all at subcellular resolution, but that's one of 4 flow cells. So we're able to get 4 times that, or 40 centimeters squared of this multiomic data per run. And that's really what makes G4X unparalleled with anything in the field. And of course, the question is, what is the quality of the data? And that's exactly what we're going to jump into next. Now that I've introduced you to the G4X platform, I've also introduced you to our multiomic assay, and I've shown you the scale of the data we can generate. I want to take a deep dive into our newest data from our multiomic assay. So I want to focus on transcript detection in the multiomic assay. I want to focus on protein detection in comparison to IF, which is truly the gold standard for in situ protein data. And finally, I want to take a deep dive into our lung cancer data set. But before I do that, I really like to highlight the way in which we did this study of our different panels and different tissues, because I think it highlights both the throughput and flexibility of the G4X. So in one flow cell, and again, remember this is one of 4 that we can run in one run, we're able to run 4 different panels, lung, kidney, colon, breast, across multiple tissue types. So let me just walk you through the layout of this run before I show you the data that came from it. So again, as I mentioned, each lane is individually addressable, has a different disease-specific panel that we introduced earlier with our curated disease-specific panels. But now we can run multiple blocks for each of those panels. So for each panel, we have 2 different cancer blocks. One of those cancer blocks run in replicate. Then we have 2 normal blocks. One of those normal blocks run in replicate. And we still have space for 2 control tonsil blocks. And we like to use tonsil as a control because most of what we're doing is immuno-oncology. You see those proteins, those transcripts very clearly there. All of this is done in one flow cell. So it's again 1/4 of a full run. We're able to do this full study, and that's where all the data you'll see is coming from just this one run. We can use the data from this single flow cell to track performance of our standard tissue types across multiple sections. In the top here, we're looking at unique genes per cell, and at the bottom, we're looking at transcripts per cell. In all cases, we have sections that are diseased, which will always be the higher unique genes per cell and transcripts per cell, and samples that are healthy. The reason why the disease have higher unique genes and transcripts per cell is a lot of those immune oncology markers are only expected to be in a disease state and not healthy. For unique genes per cell, we're getting in the neighborhood of 20 to 60 unique genes per cell, and with transcripts per cell, we're getting about 75 to 200 transcripts per cell. Of course, this level of detection is only meaningful if our false discovery rate is sufficiently low. We have 2 ways of measuring false discovery. One is negative control probes. So these are oligos that are included in the assay, but do not target any sequence in the human transcriptome. And negative control sequences that don't have a probe, but have a sequence that we read out. These are used to determine if we're having base calling errors. In both cases, the false discovery rate is extremely low, well below 1%. So even in this case here, we have relatively high FDR. It's still below 0.5%, meaning that we're very comfortable with our data quality. And this slide is one of my favorites because I think it tells the whole story of transcriptomic performance in our multiomic assay. So first, what you can see, and we'll look at colon to start, you see similar thing in breast, kidney, and lung. You see these green dots. These green dots are gene targeting probes. You can see we get about 4 orders of magnitude for sensitivity. So a pretty high dynamic range. And then down here, you see in red and in magenta are negative control probes and negative control sequences. And there's a very clean gap there. And so that's how we get that very low FDR. This reflects that and that nice dynamic range. So that shows that our transcriptional performance is very good. But in addition, what you have on this axis is the version of our assay that's RNA only. And on this axis, you have RNA plus protein. And across all 4 of these tissue types tested, you see extremely high correlation and all the dots falling on the diagonal. And really, that's just a fancy way of saying when we add protein to the assay, we see no effect whatsoever on our RNA detection. The other half of the story of data quality in our multiomic assay is protein performance. And of course, we want to compare to immunofluorescence, which is the gold standard. So we do this in two different ways. The first is we try and quantify the performance by looking at percentage of positive cells in serial sections, probed either by immunofluorescence or by our G4X multiomic IO panel. And what we're going to look at is a number of different tissues, including kidney cancer, colon, and tonsil. And we're going to look at a number of different immune markers, CD3, CD4, CD8, and CD20. As expected, on the high end, we see tonsil samples. We expect to have immune cell enrichment. In the middle, we have kidney cancer, where there may be some immune cells interacting with that tumor environment. And on the low end, we have colon. And across these different targets and the different tissue types, we see extremely high correlation, about 0.98, between percentage of positive cells in serial sections from immunofluorescence and our assay. So that tells us across the tissue section, we're getting good specificity. Of course, the next question is, what does that look like? What is the morphology? In protein, that's extremely important and brings a lot of value. And on the bottom here, you see an example of immunofluorescence from some of these serial sections of tonsil. And you see the expected sort of honeycomb, really beautiful membrane staining for CD3 in red, CD4 in green, CD20 in cyan, and CD8 in blue. And in the top row, you see the equivalent from a serial section of tonsil from a multiomic assay. And you see very similar morphology. And so what we're able to generate in this multiomic assay is effectively immunofluorescence quality data, both across the tissue in terms of specificity and with morphology. But we do it at 17-plex for protein, so you can merge these images and get this beautiful overlaid image. And this is just 4 of 17 proteins, as well as the H&E, as well as that 300 plus transcript data. And that's really what's so exciting about this assay. It's the multiomic data, the high data quality, and the massive throughput. And we think this is going to really unlock a new era of spatial biology and what can be studied. So now I want to walk you through our lung cancer data. So just to give you a sense of what this looks like, we have our 290 gene transcripts, we have our H&E, and we have our protein 17-plex. We look at a few zoom-ins here. In the protein, very quickly see some T cells in cyan with CD3+, and we see some B cells in orange with CD20. You can see that those are immune markers by the shape of the nuclei in the H&E, and we get similar transcriptional profiles. We look a bit more at the transcript performance, we see we're getting amazingly high density of detection with a median of about 200 transcripts per cell and greater than 400 transcripts per cell, the 90th percentile. This gives you a sense of, even with a 200 gene panel, the high density of transcript detection we can achieve on G4X. In addition, we're getting very nice unique genes per cell with a median of about a little more than 50 and the 90th percentile around 80 unique genes per cell. And for a panel of 190 genes, we're very happy with this performance. We're very excited about this. So you can compare protein to protein and transcript to transcript at the single cell level, and you get the expected correlations. And of course, the diagonal is one when you correlate something with itself. Now, for comparing RNA to protein, we don't directly correlate because they're fundamentally different measures, and you can very easily get spurious correlations. So instead, we classify cells as positive or negative for a given RNA or protein. Then we take the Fisher odds ratio, and we're displaying the log 2 of that value to get a reasonable scale, where red is highly correlated, blue is highly anti-correlated, white means no correlation. And finally, the size tells us the confidence that, in fact, that correlation is meaningful. So it's minus log 10 of the p-value. So a larger value or a larger circle means greater confidence. And very quickly, we can see an example of pan-CK does not correlate or is anti-correlated with essentially all immune markers. This makes sense in lung cancer, where we're expecting the disease not to be of the immune cells. You may also notice that while some of these diagonal values are highly correlated, some are not. And this is, in fact, a well-known result that transcript and protein levels for a given gene do not necessarily correlate at the single cell level. So rather than look at the correlation of a single transcript with a single protein, we can take advantage of our larger transcript panels and define transcriptional programs associated with each of these 17 protein markers. When we do this, now we get much stronger correlation with this transcriptional program that's not there when we just compare a single transcript with a single protein. This allows us to better validate our protein and transcript data against each other. We can now take that single cell data and do multiomic clustering. And so this is a UMAP based on the multiomic data with Leiden clustering based on the multiomic data. We plot those in space. You see this very clearly defined cancer region and then a healthy region around it. When we look at the top 5 differentially expressed genes for each of these populations, we get these 4 tumor populations defined by being positive for one particular gene, and then a bunch of immune cells and endothelial and fibroblast cell types that are expected with expected transcriptional programs and protein programs. So we zoom in now to one particular area in this tumor, and we see these 2 populations. In green, we have the tumor that's CD74 plus, and in blue, we have the tumor cells that are KRT19 plus. And what we can now do is take that and look at the protein level. So when we look at the transcript level, we see that, that these cells actually have somewhat similar transcriptional programs. And so in this case, the size of the dot tells you the fraction of that population that has that gene detected. And then the color tells you the level of expression. And so what we see is, for example, all these cells are CD74 and HLA-DR positive, but at different levels. So we don't really, it's hard to know what that means, what that transcriptional difference means at the phenotype level. But because we have multi-omic data, we can very quickly look at the protein, and we see that, in fact, this population is HLA-DR positive at the protein level, in magenta here, and the blue population is negative. And so we very quickly can identify the phenotype here, that these are antigen-presenting cells in this region of the tumor, in this tumor cell population, whereas in this region here, they're HLA-DR negative, highlighting the heterogeneity of this tumor and helping dig deeper into what's actually happening in the biology. We can then go back to our UMAP and do niche analysis. Now the cells are color-coded and grouped by their niche, so not just their own transcription and protein profiles, but what their neighbors are doing. And again, as a reminder, this is the tumor area. This is a healthy area. And we can now zoom in on the periphery, the transition between healthy and disease. And we see these different niches come out very cleanly. And if we then zoom in on these niches, and we can look at what's happening at the cell typing level and at the protein level. And so, for example, if we call out this green niche, we very quickly see it's enriched with B cells, and that's confirmed by the CD20 protein. And so what we're seeing here is not necessarily tertiary lymphoid structures yet, but we're getting B cell enriched immune aggregates that are likely proto-TLSs or proto-tertiary lymphoid structures. So this just highlights the value of the multiomic data being also spatial and single cell. Now, finally, I'm going to give you a sneak peek at our new RNA panels that we're very excited about. So the first is our mouse atlas panel. This is composed of 444 genes across 51 cell types. Now this panel we've just ordered essentially as a custom panel. We have not optimized it yet. So this is very new data of our first run of this panel. We ran it across a bunch of mouse tissue types, but the performance was already so good even without any refinement. We essentially see similar performance as we see in our human tissues in terms of transcripts per cell and extremely low FDR. We then do UMAP and Leiden clustering. We see our tissue types come out very cleanly. We can now look at Leiden clusters for cell types. Now, what's interesting here is we only get, for example, one B cell population. And so while most of those cells come from the spleen, if you look carefully here, you can in fact see some yellow or gold colon cells, colored cells here as well, meaning that even across the different tissue types, all the B cells are clustering together. And this is a nice validation of our assay's performance. Finally, we can see for each of these cell types, we get a different transcriptional program that's very well defined and doesn't correlate well with any of the other cell types, which is expected. We can now plot these cells color-coded by cell type in space, and we get these beautiful spatial maps of these different structures. So for example, in the colon, we see this beautiful colon crypt structure with these immune patches interspersed. With the kidney, for example, you see the cortex and medulla interactions between the two. The second panel I want to tell you about is our human pancreas panel, which has 401 gene targets. We built this panel based on evidence from the literature and existing atlasing data sets. And here you can see the breakdown of genes by tissue type. Finally, you can see the overall high-level metrics of transcripts per cell and FDR are in line with what we see for other tissue types. And we ran this panel in both in normal tissue as well as two different cancers, PNET and PDAC. PNET is neuroendocrine tumor, and PDAC is an epithelial tumor. And so, first of all, we see the cells of those tumors very cleanly cluster out. We don't see those in the normal tissue, which is good, that's expected. We also see that they have very clean transcriptional programs, differentiated from each other and from the healthy cell types you see in the rest of this plot. You also see the neuroendocrine cells basically only show up in this tumor. The PDAC cells show up only in the PDAC tumor. And finally, what I want to highlight is the expectation of the structure of these tumors. It's expected that PDAC has a very diffuse tumor structure, which is what you see here with the purple spread out across this area, whereas PNET is expected to have very high density of these diseased or tumor cells, and that's what you see here too. So this morphology confirms the phenotype we expect from these different disease states. Now, finally, because this is a human sample, we can very easily run our proteomic assay or a fully multiomic assay on it. And just very briefly, you can see the transcript layer, the H&E layer, the cell segmentation layer. But then with the protein, you can see these diffuse tumor areas with a pan-CK enriched. And with that, I'm excited to hand off to our next session with Shenya Yin, and he'll be digging even deeper into this niche analysis. I'm very excited for the data he has to share with you. Thank you.